The thallus of algae is divided into two types based on the number of cells present, that is unicellular and multicellular. Unicellular is divided into two forms depending on motility of the cell, that is, motile and non-motile categories based on the presence or absence of flagella. One motile form of unicellular are cells that can move from one place on another place, either through rhizopodial or flagellates. In rhizopodial, the cell lacks rigid cell wall and has naked protoplast, contains cytoplasmic projections known as pseudopodia and rhizophora. They possess amoeboid movement. B. In flagellates type, the cell contains one or two or many, equal or unequal and tinsel or whiplash type flagella. 2. Non-motile form of unicellular algae. A. Cacoid. The algae is usually of a spherical shape and has rigid cell wall. Lack motility, but they can be motile during the reproductive stages. B. Spiral. Unicellular with spiral filament. Multicellular form. Different or same types of unicellular algae aggregate and formed multicellular algae. 1. Colonial aggregation. In this, the unicellular form of algae loosely aggregates within a common mucilaginous envelope. They are divided into two types: a coenobium. The coenobium colony contains a definite number of cells with a definite shape and arrangement. Coenobium colony is divided into two different groups such as I. Motile. They are cellular and flagellated and can move by an organized beating action of flagella. E. Non-motile. They lack flagella. B. Aggregated forms. The aggregated forms of algae are aggregated irregularly and show a colonial mass of various sizes and shapes, divided into three groups such as I. Palmaloid. All non-motile cells remain embedded within an amorphous gelatinous or mucilaginous matrix. All cells present are independent and perform their functions individually. The number, shape and size of the cells is constant. The mucilage in palmaloid forms is secreted either by the protoplasts of the cells or it develops by the gelatinization of their membranes. Dendroid. Dendroid means tree-like. The cells are variable in number, shape and size. The mucilage is restricted only locally, generally at the base of the cell. They show polarity due to the presence of a mucilaginous thread at the base of each cell. Rhizopodial. The cells are united by rhizopodia. 2. Filamentous. When cells are divided repeatedly in a single plane and in a single direction, they form a filamentous plant body. The cells are firmly attached to each other end to end, forming a chain or a thread-like structure. It is of two types: unbranched and branched filament. In unbranched filament, they are free-floating or attached to the substratum. In the case of unbranched filaments, which remain attached to the substratum are differentiated into base and apex, whereas the free-floating one is not differentiated into basal and apical end. E. Branched filament. When a filament occasionally starts division in a second plane, they form a branched filament. The branched filament is divided into falsely and truly branched. In falsely branched, the trichome is generally fragmented due to the degeneration of an intercalary cell, after which one or both of its ends adjacent to the dead cell grows out of the parent sheath, giving the resemblance of branching. Untruly branched. The true branching occurs when repeated transverse divisions of the lateral outgrowths produced by a few or many scattered cells of the main filament. The truly branched thalli are of 4 types. They are: simple filament. The whole thallus is attached to the substratum with the help of a basal cell, and the branches may emerge from any cell of the filament except the basal cell. Heterotrichous habit. The thallus is divided into a well-developed prostrate and erect system. A system of branched creeping filament produced first and functions as the hold fast, which further give rise to a system of erect and branched filament. Parenchymatous forms. This occurs when cells of the primary filament divide in all directions. Any essentially filamentous structure is thus lost early. Pseudoparenchymatous habit. The pseudoparenchymatous is formed when one or more central or axial filaments get together with their branch fusers and develop a parenchymatous structure. Siphonocladous organization. It is restricted to members of Chlorophyceae, in which the branched or unbranched filaments are composed of multinucleate cells. Siphonous organization. In a number of marine Chlorophyceae and some Xanthophyceae, enlargement and elaboration of the thallus proceeds in the absence of septa. Nuclear divisions are not followed by cytokinesis, and the result is a coenocytic, multinucleate thallus and a siphonous organization.